Hello, I'm Shaoheng from MD Anderson Cancer Center. I'm presenting latent periodic process inference from single-cell RNA-seq data. Circular processes are common in physiological and pathological processes, including the cell cycle, the circadian rhythm, and the onset of malaria. Figure 1 shows that we use periodic functions, sine and cosine, as an activation function in autoencoder to capture the periodic changes in the gene expressions and assign pseudotime to the cells. It's like doing a Fourier transform on time series data, but the time information of the data was erased. As shown in Figure 2, the result can be used to discover circular trajectories and remove cell cycle as a confounding factor. In Figure 3, we show the performance of cyclone on a mouse embryonic stem cell dataset and 3qPCR cell line datasets. Cyclone outperforms all other methods in all the datasets. We also tested if the number of cells and number of genes affect the accuracy of the methods. We subsampled the cells and genes and tested cyclone against RNA-seq. Figure 4 shows that lower numbers of cells introduce more variances into the result, but the average performance generally holds. Because RNA-seq is based on marker genes, when the information of those genes is lost, the performance drops fast. Cyclone, on the other hand, considers the circular pattern in all the genes and can reliably recover the trajectory from as low as 2,000 genes. It's also a common need to remove the cell cycle from the expression profile because the cell cycle may overshadow other processes. For example, the cell cycle may confound the detection of the subclones in a tumor, and detecting of such subclones may be vital for a treatment. To test that, we use the MESC dataset to simulate virtual subclones. We randomly chose cells and genes in the dataset and doubled their expression to create the new subclone. We used cyclone to discover the cell cycle patterns and remove it from the profile. We tested how separated the subclones are after the cell cycle is removed. In Figure 4, we show an intuitive comparison of the separability of the subclones before and after the removal of the cell cycle pattern. The colors correspond to subclones and the shades correspond to cell cycle phases. Before the cell cycle is removed, the subclones are mixed. After its removal by cyclone, the subclone becomes the most prominent factor in the single-cell profile. The separability increased from around 0.7 to around 0.9. We also compared it with the regress out in Suret and CC remover. Both methods use marker genes. We also removed each of the principal components as a baseline. It turns out that cyclone performs significantly better than all other methods. One reason for that is that the cell cycle markers may also be perturbed in the subclones, which makes it necessary to infer the cell cycle from all available genes. In Figure 5, we show that cyclone is a metric for the periodicity of genes. We use the HESC dataset, which compares a nicotine-treated sample with a control sample. We run cyclone on each sample to capture the cell cycle trajectory and use the magnitude of the circular component on each gene as a measurement of periodicity. We identified genes whose periodicities change conspicuously. For example, the LDHA and the ENO1 are more periodic in the nicotine-treated group. Indeed, those are genes that are known targets of nicotine. They play roles in cell proliferation and also in lung cancers. There are also a group of mitochondrial genes whose periodicities are lowered. They also appear to be targets of nicotine, suggested by other studies. This metric can suggest more potential genes of interest, other than those that are up or down regulated. The transitions between epithelial cells and mesenchymal cells is vital in organogenesis, and is also hijacked by cancer to cause metastasis. It's natural to ask if EMT and MET form a circular trajectory. In Figure 6, we show our embedding of an MT dataset. We picked out the cells near the two transition points on the figure and found differentially expressed genes between them. In the result, we find a few unique MET marker genes which don't show up in the EMT gene set. This means that the two transition points may really correspond to EMT and MET, respectively, and they really form a circular trajectory. To sum up, cyclone can be used to reliably embed the cell cycle, remove it from the profile, or discover the genes whose periodicity changes significantly. There are also a lot of single-cell data analysis methods based on neural networks published in the past few years. The core of cyclone, the periodic unit, can be integrated into those tools easily. This work is published in Nature Communications earlier this year. You can have a look for further information. The software is available on GitHub.